Hi everyone, my name is Catherine and I am delighted to welcome you to this webinar today on avoiding the hype around AI and the key considerations we need to make to successfully implement AI in drug discovery. So with that, it's my pleasure to introduce you to the speakers for this webinar today. We've got Darren Green, who is the Director of Design Plus Chemistry Consultants. He spent 33 years in Glaxo Wellcome and GSK in various roles, from computational chemistry to combinatorial chemistry, screening informatics and data warehousing, the most relevant being the global head of Cheminformatics. He was driving force behind the emphasis on scale and automation of the cheminformatics systems at GSK, leading to the Bradshaw automated design platform. Darren has long been a strong advocate for the integration of computational chemistry and cheminformatics into core medicinal chemistry processes, which resulted in a collaboration with Schrödinger to develop the Live Design platform. Now retired from GSK, Darren's just started his own consultancy business, as I mentioned, and he's also Honorary Professor of Chemistry at University College London. So thanks very much, Darren, for joining us today.
Thank you very much. Yes, thank you for the introduction, and hello everybody. My first gig outside of GSK, so it's quite a moment for me.
Yeah, we're really excited as well. Next on the list, we've got John. John Overington is Chief Technology Officer at Exscientia and has over 35 years of experience in drug discovery and design, data science and machine learning. After a degree in chemistry and a PhD in protein structure prediction at Birkbeck College, University of London, and a postdoc at Imperial Cancer Research Fund, which is now CRUK, John joined Pfizer. Here, he established and led a department integrating structural biology, target analysis and prioritization, and CAD. This was followed by leadership positions at Informatica Limited's Embl EBI and Benevolent AI. Prior to joining Exscientia, John worked at the UK's Medicines Discovery Catapult, supporting the UK AI SME sector. John has a broad and extensive track record of innovation in applied pharmaceutical data sciences and machine learning and AI, spanning from genomics and genetics through to real-world data. He's got a long track record in software engineering and product delivery and has helped develop several of the foundation data resources and tools that establish the field of AI-based drug discovery. So thanks so much for joining us, John.
Well, thank you. It's a great pleasure to be here.
Do you want to say anything on this slide whilst we've got it up?
Yeah, certainly. So this is just a little bit of sort of background about Exscientia. Of course, we're in discussions at the moment, in the final stages of discussions, dependent on shareholder and regulator consent, to merge with Recursion, who are a sort of large-scale omics, phenomics drug discovery company based in Salt Lake City. But what have we achieved in Exscientia in the 12 years since we were founded? We've really had sort of encoding and automating at the core of what we do, trying to develop precision designed medicines, the right patient with the right profile and properties, and our internal pipeline and partnerships really sort of display that. We've got a number of sort of independently owned assets in the clinic: CDK7, LSD1 inhibitor, and MALT1 inhibitor, all entering clinical trial, either in clinical trials or entering clinical trials in the near term, and a number of compounds in the clinic with partners. PKC theta inhibitor is just an example of that. So an end-to-end drug discovery company, based in a number of sort of locations around the world, but very strongly driven by data, machine learning, analytics, and AI.
That's fab. Thanks so much. And yeah, looking forward to hearing more about your perspectives. So our final speaker today is Matt Segall. Matt has a Master of Science in Computation from the University of Oxford and a PhD in Theoretical Physics from the University of Cambridge. He has led teams developing predictive models and intuitive decision support and visualization tools for drug discovery. In 2009, he led a management buyout of the Stardrop business to found Optibrium, which develops novel technologies and groundbreaking artificial intelligence services to improve the efficiency and productivity of drug discovery. So, Matt, would you like to say a couple of words about Optibrium?
Yes, yeah, absolutely. So thank you very much, Catherine, and also a big thanks to Darren and John for joining us today, and to everyone who's joined us in the audience. Just a few words about Optibrium. We are a software company focused on software and AI solutions for small molecule design, optimization, and data analysis. Probably best known for our Stardrop software, which is a complete package of fully integrated and elegant software for small molecule design, optimization, and data analysis. Sorella is our AI platform that uses great methods like deep learning imputation to gain new insights from the very complex, sparse, and noisy data we deal with in drug discovery to improve the efficiency and productivity of drug discovery. Biopharmics is our platform in 3D molecular modeling, really scaling from small molecules to large and complex macrocycles. And Symmeta is our sort of unique mechanistic approaches for predicting phase 1 and phase 2 drug metabolism. So a very broad platform. And yeah, if you'd like to learn more, you'll see lots of links on the slides there. So thank you very much, and look forward to a good discussion today.
Thanks, Matt. And thank you all for joining. As you can see in the overview today, we've got quite a lot to discuss, lots of points to get through. But we'll also be taking some audience questions as well. But before we begin, I thought it'd be useful just to set the scene on the current state of AI in drug discovery. So, as we all know, drug discovery takes a long time and a lot of money. AI-driven discovery is on the rise and promises to reduce these costs and the time taken for discovery. It's more frequent now that we hear stories of remarkable speed to clinic powered by AI, and it's a really, really exciting field. However, there does tend to be an awful lot of hype around some of these AI methods. So, for example, maybe things don't reflect accurate drug discovery data sets in real world scenarios, some of the examples. And there can be there's a lot of skepticism out there about AI, and significant barriers still exist for adoption. As you can see from some of the stats here, it is growing significantly with the highest average adoption rate in the small molecule sector and the highest annual growth rate in understanding diseases. So with that setup, it leads me nicely onto my first question to the panel, which is, where do you think AI holds the most potential in terms of improving drug discovery? Which areas can it tackle most effectively? So with that, I'm going to stop sharing my screen so we can all focus on your lovely faces. And who would like to go first? Darren, perhaps, would you like to give your first thoughts?
Hey, I'll try not to mention all the areas. I'll just pick a couple, and we maybe can go round. Obviously, you know, my core expertise is around generative design. I do believe that that's an area, when coupled with good machine learning models, I do think that's going to have significant impact. Quite remarkable to see the results of that survey actually in small molecules. I mean, QSAR has been around for forever, and you know, most QSAR models these days are using machine learning, and you know, to about, was it 50-60% saying limited or no impact? Wow. That's very surprising, because you know, those models should be having an impact on how people work. I think there's more than enough data out there to show you that these models are good enough to do these things. But I think that the sorts of examples we're seeing primarily from the startup companies, Exscientia, in silico medicine, you know, Schrodinger's application arm, you're beginning to see the efficiencies that I think we can achieve if we use these techniques properly and integrate them correctly. And the other one I'll pull out actually is probably not non-scientific, but I guess more generic to the workplace, in terms of, you know, the impact maybe large language models can have on literature searching and summarization, paper summarizing. you know, particularly document writing. Anyone who's seen, you know, the truckloads of paper that I think can still be sent to regulators when you actually, you know, submitting all your evidence, and the extremely long amounts of time and large numbers of people that are involved in writing such documents. So you can see things like large language models. I think we'll definitely see an impact there. And you know, I think we do tend to focus a little bit in our world on the you know, sexy molecule generators and things, but I think there's a ton of core stuff that you'll see companies just make us more efficient. And if you can, you know, halve the time taken to write one of those documents, you know, that's actually as good as knocking 2 or 3 cycles off your lead optimization. You know, might be a lower hanging fruit, to be honest, in terms of getting going as well. So I'll stop there, let the others chip in.
That's really great. Thank you. Yeah, and it's definitely important, freeing up more time for science. John, what about you? What are your thoughts on this?
Yeah, so I think, you know, as Darren and many others have sort of highlighted, the generative AI is now a pretty, actually, I think, a mature technology. Generating molecules isn't really a problem. Constraining them to be synthesizable, or in particular scoring them against ever increasingly complex endpoints now becomes a challenge. So I think better predictive models is another area of great sort of future progress. Automation is really core to some of the things we're doing. There's a little point being able to design a compound in 7 nanoseconds if it then takes 5-6 weeks to order from 6 different vendors and so forth. So I think this really accelerating the design cycles is sort of key. One of the other great sort of frontiers is I think most of the current generative AI approaches, certainly not all, are essentially steady state. So a compound in a system with a defined number of components. I think turning these things into the time domain, so the pharmacodynamics, the pharmacokinetics, the toxicokinetics, and so forth, is a big challenge. And I think, you know, they're really hopeful to see some big improvements. You know, just through conferences I go to, start to get really impressed with some of the things that people are doing. But it's a fast-moving field. Staying on top of stuff, the literature is just very, very difficult now.
Yeah, that's true. Matt?
Yeah, I mean, I think that that Darren and John have really touched on some really key points from a chemistry perspective. I sort of think of it as the sort of triumvirate of AI in chemistry, which is, you know, the generative model saying, well, what might we consider making? The predictive models that say, well, which ones should we be considering making, which are predicted to achieve the balance of properties we need in a safe and efficacious compound for our therapeutic objective. And then which of these molecules can we make in a reasonable time? So the retrosynthetic prediction, or linking that through to the available chemistry and so on. And so that's an area we very, very much focused on. But there's another element as well, which is not just about what new molecules to make, but the decisions we make in the progression of molecules through our projects. So ultimately, we're looking for high quality compounds in very complex downstream endpoints, whether it be, you know, phenotypic sort of secondary screens, or even in vivo, whether it be efficacy or pharmacokinetics, as John mentioned, and leveraging the data that we have in early drug discovery to make better decisions about which compounds to progress by more accurately predicting those downstream outcomes using, for example, the sort of imputation methods that we have within our Sorella platform is really important. But then bringing in the element of active learning. So using the models to tell you what is the most valuable data to generate in order to make better decisions about progression downstream. So maximizing the return on investment in our sort of experimental efforts, as well as in what new molecules to make. Obviously, we've been talking a lot about chemistry here, because I guess particular areas of interest for us. But Darren, again, I know you've got a much broader experience in applications and more in sort of biology and other spaces. I don't know if you had anything you could sort of point out from those areas.
Actually, I'm very excited by protein-based applications, because I think obviously, peptides are more of a natural text-based, string-based representation than a chemical graph. And I think you've seen some quite extraordinary examples from the Baker Lab, for example, recently of, de novo protein design, tested in the lab, taken all the way through. And so I think, those areas are, fascinating. The thing we haven't talked about really is target ID, where, you are seeing, this whole thing of literature scraping and evidence ranking and assembly and presentation to a scientist, saving time, but systemizing that, taking the bias out, being able to integrate literature with omics. I think there's some nice examples coming out of those sorts of things. Obviously, it's a lot longer time scale on a readout of target IDs to whether you've really made a difference. But I think you can get some interim measures on, is doing something in that target, give you the phenotype you want and things like that. So yeah, I mean, that's another area that I think you're going to see a very large amount. I mean, John, the Exscientia paper on precision medicine, I thought was fantastic example of impact on patients that we can see happening.
Yeah. So yeah, just to sort of build on a couple of themes from both Darren and Matt. So probably the majority of the audience are interested in the center section of drug discovery, and primarily small molecules. And there's other things going on around that. The biologics is certainly one. A lot of clinical AI. So getting the right drugs to the right patients, using omics or biomarkers and so forth to do that. And that's a great thing to do, but you really need products to make money from that, or you need to work with people with products to make money. And as a general comment on the target discovery side of things, I think evidence is showing that's, but because of the long time to value, that's quite a difficult place for a small company to sort of monetize. If you look at a few years ago, 23andMe, with all the genetic data that they had, the potential to use this as a great resource in target discovery, and now, as probably many people know, certainly their customers who may have had their data breached, which includes me, it's tough to make money from that sort of thing. So I think this community's in a sort of sweet spot. I genuinely think the small molecule area has got a lot of really tricky, gnarly problems still to solve that are within reach. The biologics, again, in the sort of the market, very large number of startups. It's easier to do gene synthesis and expression on proteins. You don't have anywhere near the same. It's not to decry the level of skill and judgment for that field, but it's fundamentally simpler. And I think it's going to be a tougher place to actually come up with commercial success, purely because of the large number of companies there long term. It's exciting time.
I was going to say that leads really nicely into the next question, which is about where AI might not work. So, this webinar is all about avoiding the hype. So are there any areas of drug discovery where you think it's not worth using AI? John, would you like to start on that one?
Yeah, so it would be great to do something transformational with AI in the sort of toxicology space, and clearly a lot of people are working there, but arguably we do as an industry a pretty good job of getting rid of a lot of toxicity through conventional screening and a whole bunch of models that have been around for some time. The sort of tox that kills us now is, not literally, of course, that's a bad word to use. The tox that kills programs, stops them, can sometimes be just idiosyncratic or mechanism based, and that's quite a problematic thing to find. So I see toxicology as being predictive AI toxicology as being like a great thing to aspire to, but it's a tricky thing to do. Would people really believe the models? Would they take decisions on the basis of it? Are they ever going to be predictive enough to find 1 in 10,000 events that could kill a compound? So it's challenging.
Really interesting. Darren, do you have any thoughts on this?
Yeah, so full disclosure, I'm on the last SAB, so they deal in computational toxicology, and I think I think where you will see things play in that space is, what am I seeing in my animal experiments or human experiments? Can I understand what might be causing that? Can I then translate to, okay, it looks like we're hitting this pathway, can I monitor that? Is that reversible? Again, come back. Can it influence my decisions or my plan for how I'll develop this drug? I think in terms of hype, I think people get a little bit carried away. We see examples, and we think, "Wow, great, that's good." And so benevolent repurposing that COVID, I wrote it down because I'd never remember it. Baricitinib. Fantastic, right? But if you do a Google Scholar search for drug repurposing in COVID, you'll get about 20,000 papers. So, is that repeatable? Is that just an exception that proves the norm? So I think we when we publish and when obviously people get successes, you see those out there. Well, what about all the failures that we're seeing? So to get a true picture on the performance of things, one has to experience it across a portfolio of applications and projects. And so it's a little bit hard to know the true performance on things. The thing I was going to pull out is, working on things that are outside your training set. We're bedeviled as an industry, and we talked about tox, small data sets, sparse data sets. And so, we're still not at a place where we're great at using small data. Yes, all the hype around AI comes from having large data. Plus, your data's got to be relevant to the problem at hand. And if the classic thing in chemistry, we're always going outside of a training set. That's because that's what we do. And so that is still the challenge of how do you quickly get the right data that will help you build a good model that can, because I think I've seen enough examples of where you've got really good models, all this stuff works, right? The challenge to me, as John said, how do you score them? And therefore you are going to need either a simulation to generate the data to generate your machine learning model, or you're going to have real data. And how do you get that quickly, to pull all those things together and efficiently?
Just as a follow-on to something Darren just said. So, we're still fundamentally data poor, and good data is expensive. So Kemble's been quite a transformative resource for training a whole bunch of generative models and scoring and so forth. But if you wanted to build Kemble, I was doing a sort of sum for EuroQSAR where I gave a talk last week. You'd be looking at between $30 and $50 billion to regenerate a data set of the diversity and relevance of Kemble. And that's outside the reach of everyone, really. Drug discovery data is expensive, especially the late stage in vivo type data and the stuff we're now really sort of struggling with. So data is a big issue. And you can't just synthesize that type of data as well. Some data is within the reach of synthesis, high quality molecular geometries and things from first principles. But some of the real challenge data is just fundamentally hard work and a lot of money.
While we're talking,
Oh, sorry, Catherine.
I was just going to say, while we're talking about data, we've had a really good question come in from someone about when AI is trained on public data, whether there's a problem about bias towards positive findings, because obviously positive findings are what tends to be published, and how that influences trust. So I don't know, Matt, would you like to comment on that one?
Yeah, so I mean, I think one needs to be careful with public data and for several different reasons. One of which you're quite right is that it does tend to be sort of biased towards, you know, positive results. If you look at the distribution of activities in Kemble, for example, they tend to be biased towards the more active molecules because they're the more interesting ones to publish, for example, in a MedChem paper. So yes, one has to be careful about bias, and there are various approaches you can take to synthesizing negative results, for example, or and synthetic data, as John was saying, and so on. But probably even a bigger problem is the noise in that data as well. And there's been some sort of great papers recently looking at the sort of reproducibility of those the correlations between results from different assays ostensibly for the same target and the same activities, and just the amount of noise that introduces if you try to sort of bundle all that data together and build a model. So yes, public domain data can get you a long way, but one has to be very, very careful about, you know, curating that data for your specific purpose and making sure that that you're not sort of trying to model the noise, if you like.
That's great.
I think reaction prediction is a classic example of that. I think that's why we are where we are with reaction prediction. I had that on my list of hype. It's like we seem to be stuck at a particular level because the literature is just publication bias, and you really need all, you know, journal of failed reactions or stuff that didn't quite do what I thought it would do. Yeah.
I think there's another important point here, which is, you know, AI is not magic. I hate to break it to everyone, but we actually know quite a lot about chemistry and physics. And expecting sort of AI methods to sort of magically learn all of the sort of underlying principles purely based on data is, you know, given the amount of data that we have, as we've just said, is probably unreasonable to expect. You know, one example of the hype around it, there was a huge amount of interest in this, the DiffDock method that came out of MIT, I think it was, and really getting some quite remarkable results in terms of the sort of RMSD in prediction of binding to protein targets. But actually, I think it was a great paper by Charlotte Dean and her group at Oxford on a method called Posebusters, when he actually looked at the actual structures that were being generated. They were just, you know, physically, chemically impossible. There were some beautiful examples where there were two aromatic rings that were linked like links on a chain, which you know is just not plausible. So, you know, you give an AI method a metric to optimize, it will optimize the heck out of it, but it doesn't mean it's actually captured the reality of chemistry and physics. And we shouldn't forget about, you know, the good old-fashioned methods that actually, at least empirically, encode a lot of those rules, whether it be, you know, scoring functions and docking models actually do contain a lot of prior information that's been learned over many years. And perhaps one approach to, you know, making the most of the limited data we have is to build methods in which the chemistry and the physics is explicitly encoded, whether it be in the descriptors or the inputs to the machine learning. That enables us not only to learn based on less data because our descriptors are much more information rich, but also to extrapolate beyond the small data sets we have because those inputs are based on, you know, real chemistry and physics.
Yeah, just picking up on the word hype, I guess a few years ago, for me, something that doesn't appear to have delivered what the community and champions suggested was around federated learning. You know, it just doesn't seem to have caught on in a way. Either there isn't enough data and the diversity of it to actually share, but you know, the experiment was done. Maybe it's been done a couple of times. And so forth, and the community sort of learned from this. But yeah, we got to, yeah, it's an area of high innovation and risk.
Yeah. So I guess, yeah, my name's on the Melody paper, so I agree with you. It's an experiment we had to do. I I think with federated learning, there are lots of things that one could do that we couldn't do in the Melody framework. And I guess the challenge is with the importance of, and John, mentioned the cost of generating Kemble. I think everyone now is much more aware of the value of their data. And so I guess the things that you would probably need to do to go do a better experiment than we were able to do with Melody might not be agreeable to companies now they understand the value of their data and the potential risks of that data leaking to do a better federated learning experiment. So maybe it's not the technology, it's the fact that we're just not willing to throw it all together in the open so we can use the best methods we possibly could. I don't know.
This has all been super interesting. I'm going to move us on to the next question, just for time. So we've discussed a lot about where AI can be applied and where it can't be applied. But how do we prove that AI is actually providing good value? How do we make sure that we're getting significant return on our investments and useful results? So several questions in one there. But Matt, would you like to start us off on this one?
Yeah, absolutely. I think there's a tendency amongst scientists to look at the stats and to dive in and say, oh, is my R² better than this other method? Or we talked about an example a minute ago, is my RMSD better in predicting poses of compounds? But yeah, this sort of battle of the R² really doesn't demonstrate value at all. It's great for sort of academic interest, and it's great to sort of prove the concept, if you like, but what really matters is the number of compounds we need to synthesize to get to our goal, the number of experiments we need to do to find those high quality compounds, and the time that it takes to get there. So that's what really drives value for drug discovery. And I guess the other thing, when we look particularly at retrospective analyses to try and demonstrate the point, is also not just can we get to the same point faster or cheaper or better, but have we missed anything? Are there new opportunities that would have been identified by using these AI methods that could be even more valuable than the ultimate outcome? But these are things that really will ultimately drive the efficiency and productivity of drug discovery.
That's great. Darren, would you like to add something?
Yeah, I mean, I sort of touched on it earlier. I think it's being able to repeat it. So it does. It's a maturity thing. I think John mentioned the maturity thing. That's a thing of maturity. Yeah, I mean, measuring the right things. Matt touched on it, but I think for small molecules, it's an economic argument. Small molecule discovery is expensive compared to biologics, and there's an economic angle there that you've got to measure this thing. And I think of these things in terms of time and money, rather than number of compounds made, because you can make it in an array, it's cheap. So not counting compounds so much, but number of people you've got to commit to this, the expense of doing it. Really be interested to hear John's thoughts on this, because a small company, I imagine you think about this quite a lot, whereas in a large company, you've got a DMPK department that they're waiting for your compounds to come through. I think the other thing is be clear what AI is, right? There was a review paper, I think Boston Consulting Group published a paper, AI drugs coming through, and if you actually go and look at the things they quoted as AI, it's probably 90% of them didn't really have any machine learning in there at all. So you've got to be clear about what we're talking about and the contribution of whatever that technique is. Because actually a lot, when we were at GSK and Bradshaw, more than half of that platform is just large scale it's large scale cheminformatics. It's not AI. It's just good stuff. Yeah.
One of the challenges that I occasionally get around the use of AI in chemistry is people expect the compounds to look really novel, unlike anything ever seen before. And as if this is yet another hurdle that AI needs to sort of get over and things. And for me, you know, a brilliant thing would be a Turing test that experts couldn't tell whether a compound was designed by an AI algorithm supporting humans or through traditional humans working through graft and experience and things. So a Turing test for design, you know, to expect a novel target from a company, novel mechanisms of action for an untreated disease with a compound that looks like nothing else ever against a target that's historically been undruggable. Can you imagine a more unicorn-y unicorn than that? And if that's what the hurdles are to sort of measure the impact of AI, we're never gonna get there. The stepping back and picking up on something that sort of Matt mentioned is benchmarking. Yeah, we spend a lot of time benchmarking methods, and very few papers actually have a benchmark result that's worse. Yeah, we spent a lot of time working on something and we're worse than all the other methods we've previously troubled. So I think there's a reproducibility crisis brewing around benchmarking in this field. We certainly take external, nominally very well benchmarked or benchmarking tools, try it on our data, which is blind and things, and it you know, they nearly they invariably never work as well as in the publications, and this, you know, in a way slows down the progress of science, slows down adoption. So a great fan of some of these competitions and you know, sort of third-party independent sort of you know, critiques of benchmarking and things. I think, you know, they're a great thing to do, but the literature itself chasing, you know, a 0.02 increase in an R squared or something, or an AUC, you know, it's tricky to do.
That's a really good point.
Yeah. And I think just to pick up on, I guess, points that both Darren and John made as well is, you know, proof is a difficult word, and particularly statistical significance of that proof. I think we have to accept the ideal experiment would be if, you know, we ran the same project twice, one with AI and one with old-fashioned medicinal chemistry with no support from computational methods, and we saw how fast they got to the goal and the quality of the goal, what they achieved, and so on. No one's going to do that experiment in practice. And secondly, even if they did once, they're not going to do it 50 times that we get a statistically significant result. So we do have to accept that you know, a lot of these proof, if you like, are going to be anecdotal, but an accumulation of anecdotes hopefully gets to at least convincing, if not statistically significant evidence.
That's really great. Moving on to our next question. What can we do to gain maximum value from our AI? So encouraging more users, driving adoption from that side of things. Is there more that companies could be doing? John, would you like to kick us off on this one?
So it is getting adoption even within companies can be quite tricky. Actually, it's a problem faced by many many organizations. People are familiar and comfortable with the tools they have. Using novel things can be stepping out a little bit into the dark, and humans naturally love to be in their domain of applicability. So I think there's a human issue in motivating and incentivizing people to try new methods and fully swallow the Kool-Aid, as it were, you know, which is the other end of the sort of spectrum. But adoption is key. We had a corporate goal last year specifically around adoption of our tool stack. You know, we had, of course, you know, we had some conventional tools and some internal AI tools and so forth, and actually started to track and measure this. We've been sort of very pleased with the progress, but it's only once we started to measure the adoption and the use that, you know, patterns became clear, really helped direct some of the areas that we needed to improve, and also some of the areas that were perfectly good enough and we had fantastic roadmaps for many years on, but they were good enough already, I guess, is one way of thinking about it. So measuring performance of systems and adoption, I think, is a key thing.
That's great. Who wants to go next, Matt?
Sure. I think there's a couple things here. One is sort of robust deployment of these algorithms and methods. It's great to have a really powerful algorithm that your expert data scientists can sit in the back room and run from the command line and generate marvelous results if they've carefully prepared the data and done a lot of sort of manual tuning, all of which takes a lot of time and effort. But ultimately, to gain real value, it needs to be implemented in a way that is not only accessible but robust, that will deal with the edge cases where the data isn't quite perfectly formatted and isn't quite ready to go, so that anyone in the organization can run it with confidence it's going to give them useful results in a timely way. And then John's already touched on accessibility. Again, it can't just be in the hands of the super experts for a couple of reasons. One is there aren't enough of them to go around. It's that balance between how many computational chemists do you have per medicinal chemist, and typically the computational folks are heavily outnumbered. And secondly, it is that sort of instantaneous response. You want these in the hands of the decision makers. So if they have an idea, they can say, oh, what does this look like? Or they can instantly assess the output of a generative method and so on. And it's that sort of feedback loop that really enables you to get the most value from it. And so, yeah, accessibility to non-experts and deployment in a really robust way to make sure that it generates reliable results.
Thanks. Darren, do you have anything to add?
Yeah, I can see we're going to have an interesting discussion on the next question, actually. I think the thing that we found when we were doing our first large-scale Bradshaw project, how difficult it was to shape the output of these very large-scale experiments for decision making. And I had not anticipated how hard that was to make it digestible. That you've looked at hundreds of thousands of molecules, and we're going to talk about these 50. Why are we talking about these 50, right? And what about them, and this sort of thing. So that's really quite interesting. Can have all the clever algorithms in the world, but at some stage a human's going to digest it, and how do you shape it? Which I think is fascinating. The other thing I think I would say is actually thinking about how we look at software systems now to exploit this, pushing things to people, not saying you've got to click these 15 buttons to get an answer, but actually based on the data and the system knows what you're trying to do. Can the automation of this and pushing the right recommender systems or whatever they are to people, I think that's we've got to start doing that, because you don't want to have 10 people all pressing the same 15 buttons all the time to get the same answers. That seems a bit silly. So.
That's great, thank you. We've only got one more prepared question, and then we've got an absolute ton of audience questions that have come in, which is fantastic. So we'll whiz through our final prepared question, which is about the organizational perspective on increasing our value from the AI that we're using. So what do we need to do on an organizational level to ensure teams can gain maximum value? And Darren, you already mentioned that this was going to be some interesting discussion, so I'd love for you to kick this one off.
Yeah, sure. So as a card-carrying computational chemist, Cheminformatics, Matt mentioned, too few computational chemists in companies, or and that ratio has to change. Full stop. Because this world now, if a company is going to exploit these technologies, you need more specialists. So for a large pharma company where you don't have that heritage, there's a challenge there that needs to be addressed. Obviously, for small startups that are based on this stuff, then there's not that problem. It's really interesting. You look back in the past, structure-based drug discovery, Vertex was formed to do that properly. Fragment-based drug discovery, Aztecs was formed to do that properly. AI drug discovery, Exscientia in silico medicine were formed to do this properly. I think we should add, I don't know what you call it, dynamics-based drug discovery, what Schrödinger are doing. I mean, but they're doing it properly. So I think it's a really interesting thing. In a large established company, you have very large change management issues. The smaller company, you can sort of design your company around the approach. But it's fascinating here, John. It's like change management challenges aren't only in large established companies. So,
oh yeah, no, they're everywhere. It's a really interesting point around just digging a little bit deeper into Darren mentioning Vertex. So the companies I really admire, Heptares, or what? I just can't remember their current name, but I think the really successful platform companies, in my view, have largely been structure-based. So they've had structure at the core, and they're a little bit broader than just cheminformatics. They're the interface between the biology and the chemistry. And I think structural biology is a great unifying sort of feature and technique. You've got to understand geometry, you've got to understand molecular structure, you've got to understand interactions. And this duality of working with the target and the compound at the same time, I think are really where the action is. And that's where, I think the sweet spot for designers are and so forth. And just picking up on that again a little bit, some techniques that were arguably ahead of their time, like Comfer. We didn't really know how to do molecular overlap and so forth. And this was back in the 1990s, maybe even before that. And imagine re-looking at that now through the lens of the type of data we have, the ability to do co-folding, docking types of things. And say, I think there's so much opportunity, but it's this interface between the biology and the chemistry, ideally unified by structure. And all those companies have been remarkably successful at discovering drugs, real drugs, that are now impacting patient lives and things. But they had a core of just a vision, a technology vision, a platform, a scalability around their sort of skill set.
I might just sort of maybe challenge a little bit what Darren said, in the sense that, in an ideal world, we'd have organizations that were all built around the principle of using AI and make sure that they had enough computational chemists and data scientists to support all their projects. And organizations like Exscientia are of course designed in that way and are designed to gain that value. But I do think you can go a long way by making sure that the methods that are available are accessible and enabling and augmenting the capabilities of very skilled and talented medicinal chemists. I think that there's a quote I love to take from Alpha Lee from Postera, who basically said, AI is not going to replace chemists, but chemists using AI are going to replace chemists that don't. And so, really, you don't need an army of computational experts, provided you can make these methods interpretable, make sure the results that are intuitive and can be used within the decision making. So, I think that the balance is important there. But overcoming this sort of reluctance, if you like, is really important in understanding that it does require organizational change. That you'll be making molecules you would not normally from a medicinal chemistry perspective. You'll be running experiments that you wouldn't normally do, because the information being generated by those experiments will, if you like, sort of feed the beast of the AI. It's providing that information that it can learn from to ultimately make much faster progress. And the number of times you say, well, we need to make this molecule to get some data. I know it's going to be inactive, or it's very likely to be inactive, but it's going to gain us some really important information on sort of metabolic stability or toxicity or whatever it might be. You often get a reluctance. Well, I don't want to make an inactive compound. What's the point of that? And the point is it provides the information that we can sort of grist to the mill, if you like, to ultimately design better compounds using these computational methods.
Thanks, Matt. I think that leads nicely into one of our audience questions. about when you train AI on a defined chemical space, is it realistic to expect innovative solutions in a novel chemical space? So I think, Darren, you first brought up generative chemistry. Would you like to answer this one?
Yeah, it's well, yes and no, I guess, is the answer. And it comes back to, I forget who mentioned it, but what has your model learnt? So has your model been able to generalize? Really important for the model to be able to tell you about when it's uncertain, why it's uncertain, and generally you can normally go outside the training set. You can't go too far, but it's important to have the model tell you that, not looking at the structures yourself, because the model knows better than we do how it's uncertain and whatever. But it's important to do that, and this is where Matt mentioned active learning and so on. So this is the embracing of those optimization techniques that are proven to be more efficient than humans at doing these things, right? And so coming back to a last question around the sort of adoption and culture change, have to embrace these things, because that is how you'll get the best out of the approach.
That also ties in really nicely to another question that we've got. So you've mentioned we have to embrace AI. Someone has asked whether, based on the dataset sizes of most biotechs, should we be encouraging AI uptake? Because in many cases, there isn't enough data or solutions for to really stretching the budget. So whichever one of you would like to answer this question. John, maybe?
Yeah, I think clearly building an AI data platform in the context of a small company who's primarily trying to develop a product drug is probably not a good idea. The money is not going to be available for that, though. I think leveraging open source models, web servers and stuff is a great thing to do, as long as they're secure and protect IP, though. So it's embracing in the right direction and context for a given company, I think, is the answer. So it's good leadership, it's good management, and it's good strategy that are behind that. Yeah, certainly a sort of just rushing into generative AI for the sake of generative AI, or to make some CEO or senior manager happy, and is probably not going to be a good recipe for success.
Yeah.
I also think it's about the right tool for the job. There are some AI methods that that are only really suitable when you've got lots and lots and lots of data. So one classic example of that, in my experience, is sort of the graph-based neural networks for predictive modeling have been shown to offer really quite substantial improvements in the quality of predictive models, if you have a lot of data for which it to sort of dissect those relationships between graph structure and the predicted properties. But in the majority of cases, which is the reality where we don't have tens or hundreds of thousands of data points, actually they add very little value over good old-fashioned random forests. And so you don't need to get the very latest, cleverest methods. Some of the more conventional sort of QSAR methods are very, very good indeed, with relatively small data sets. And it also ties back to the point I made about the types of descriptors and the way we characterize our molecules as input to these models. The more information rich those inputs are, the less data you need to recognize what makes an active compound or an inactive, or a permeable compound from an impermeable, whatever you're trying to model. We need more data where there's a more tenuous link between the structure of the molecule and the property we're predicting. And so using these much more sort of information-rich descriptors that are linked back to the chemistry and the physics of what's going on is really important. And that actually ties back to the last question you asked as well, about expecting things to sort of work in new chemical space. And again, by linking it back to the chemistry and physics, again, sorry, same words, but that enables us to extrapolate in chemical space much, much better. And there's one very nice example that two of my colleagues, AJ Jan and AJ Jane and Anne Cleves published with Corteva very recently, where by using a model based on actually modeling the surface properties of molecules and active learning, as Darren discussed, showed how it could very quickly move from a model initially built on a series of macrocyclic compounds that were, synthetically very, very difficult, into predicting highly active compounds in a fully synthetic series. So a completely different chemical space, but leveraging that information very effectively to move from that macrocyclic series to a synthetic series very much quicker than proved to be possible originally in the project.
Yeah, and that's a great paper. People should go and have a read if they want to. I think it's on our website. We've just got a few minutes left, so let's pick it up with the questions. So the next question is, should AI or machine learning be focused on designing novel scaffolds, or more focused on R groups, which are typically easier for chemists to synthesize? So yeah, where should our generative chemistry efforts be focused? Darren, would you like to take this one?
I think I think you have to couple it with the models you're using. So if the model is sort of saying there's a problem with this, with the scaffold, DMPK or whatever, then so the generative chemistry should serve the objectives you are trying to get to and the problems you have. So and actually for some cases, generative chemistry might not be the best thing to use. You might want to use a classic pharmacophore or based replacement or a bioisosteer replacement type of thing. But so I would turn it around, always look at what you're trying to do, and then what's the best tool to do that for me.
Great.
Sometimes the issues with scaffolds are sort of fairly fundamental to do with, say, metabolism, cytometabolism, or liability, but often also they're patents. So you're getting patent novelty in an otherwise excellent molecule. And again, I think generative AI scoring and filtering and the inclusion of IP space is absolutely essential as well. You need a patent. You need to be able to protect your molecules. And yeah, the number of examples where just single atom switches have both transformed the safety of a molecule, but also the patent position is just never fails to sort of amaze me.
And that links really nicely to a question that someone's asked on whether AI and LLMs could be used to write better and more comprehensive patents. So I think you've already sort of touched on that, but do you have anything else that you'd say?
I think that is going to be, an absolutely incredible time and actually revenue-saving part. Filing patents quicker is going to be an issue. One of the surprising things, though, is that patent analysts and patent writers are one of the people that sort of tell me, at least, that their jobs are completely safe from AI and so forth. So there's always going to need to be a human in the loop, which is certainly true. But I think in terms of the drafting, the tools are already there, to be honest.
Yeah. Cool. Well, with that, I think we've just about run out of time. So I'd love to thank all of our speakers, and I'll just leave you with one final screen, because we will be talking more about AI methods in just 2 weeks' time, or maybe 3 weeks' time. So would love everyone to join that. But yeah, all that's left for me to say is thank you so much to our speakers. There are loads of questions left, so I think we'll need to get you back on for another webinar from the sounds of it. Yeah, thank you so much. This has been really insightful conversation. And thank you to everyone for joining us as well.
Thank you so much.
Thanks,
really
Captain.
enjoyed
Thanks,
the discussion.
Darren. Thanks, John.
Yeah. Thank you very much. Yeah. Thanks, everybody.
Bye.
Bye-bye.
Bye-bye.